Today I will show you how we can work with Motor-Imagery BCI dataset, in particular the EEG dataset acquired from imagined limb movement experiments. As we know, our movement is controlled in the motor cortex, where there is an increased level of mu activity between 8 and 12 Hz that can be observed in EEG data, okay, especially when we perform movements. We can observe a reduction of such mu activity in specific regions of the brain that deal with the limb that is currently moving. Such decrease in EEG mu band is called event-related day synchronization, or ERD in short. If we measure the amount of mu activity at different locations on the motor cortex, it is possible to determine which limb the subject is moving. Through mirror neurons concept, this effect also occurs when the subject is not actually moving his limbs, but imagining it, merely imagining it. This is a very important finding for BCI. So whenever we imagine limb movement, we can observe mu activity suppression in EEG data around 8 to 12 Hz, also including the beta region, 8 to 25, 30 Hz actually. So today we will work with one of the popular Motor-Imagery BCI dataset that is available online as a part of BCI competition. The dataset for this tutorial is provided by the fourth BCI competition, which you'll have to download yourself. For that, you can go to the following website, fill in your name and email address. You should receive an email that contains your username and a password for downloading the BCI dataset. Make sure you download the dataset 1 from Berlin, okay, with 100 Hz sampling frequency, let's say in MATLAB format, as shown here. So this is the link where you need to go to download. And then you should unzip, create a directory, subdirectory called dataset, data_set_4, and it should be placed inside the directory in which you have stored these files. Okay. All right. So yeah, the description of the data is available. You should read it in detail on the following link. Now let's start loading the data. So we import NumPy, okay? And then since we're dealing with a MATLAB file, we can use SciPy's input-output function to load it. All right, so import NumPy, import SciPy IO, for example. So SciPy IO load mat allows you to load the dataset. This is one of the dataset we have, okay? So once we run this line of code, M will be the object that stores our dataset. So this is what we obtained. It's a dictionary type object. M keys. Okay, so M keys should give us the detailed information. Header, version marker, CNT, right? And SciPy load mat. Okay, doesn't deal well with the MATLAB structures, which result in lots of extra dimensions in the arrays. This makes the code a bit more cluttered. So we can somehow extract the information related to sampling rate. Okay, located here under M. Okay, let's in NFO, FS frequency, sampling frequency, and EEG data is located at M CNT transpose. I think the CNT stands for continuous data. So if we run this, we're going to obtain the number of channels, number of samples, all right, this month, and then the sampling rate. Look, this is 100 Hz. So the data has been sampled using 100 Hz. So we can further extract the channel names information and then other marker information. So channel names. Okay, now we have channel names. And we can quickly take a look. So these are the names of the channels. In total, we have 59 channels. right? And then event onsets. So event onsets. Event onsets will be used for segmentation, right? So these are the onset, the time samples where these, for example, trigger information has happens. And event onsets has, let me clear this out, event onsets has event codes. These are basically the markers, right? So you see, event codes encode binary classification tasks. So one, for instance, is left-hand imaginary. Okay, -1 is right-hand imaginary task. So event codes along with event onsets can allows us to segment the continuous state set. Event codes, for example, what's the shape? So we have 200 samples, right? So now we can further load, let's say, labels, okay, and then, okay, so right hand and left hand imagination, okay, classes to labels. So CL1, CL2, right? So two class classification tasks. 190,473, sorry, 190,473 labels. So now if you just print all the information, we have, okay, the following. 59 channels, sampled 100 hertz, okay, and then this amount of time points, channels, events, number of events, event codes minus represents left, okay, plus one represents right class. So binary classification task, two class. We want to discriminate between subject imagining right hand movement and then left hand movement. This is a large recording which has 59 electrodes spread across the entire scalp. Okay, if you go to the website, to the description of the dataset, we can find more details. So if I open up this, okay, this is the dataset description. All right, so experimental setup, we have calibration, evaluation, special feature. Okay, what we want to find out is the experimental paradigm. Okay, these datasets were recorded from healthy subjects. Okay, in the whole session, Motor-Imagery was performed without feedback. Okay, for each of subject, two class Motor-Imagery were selected in the three classes, right? Left hand, right hand, and then foot. As we can see from this cortical homunculus map, the foot movement is controlled at the center of the motor cortex. This is the motor cortex, right? This is a homunculus map that we know. While the hand movement is controlled more lateral. So this is for left hand movement. Okay, location of the brain area responsible for left hand movement. While the right hand movement, as we know, is again contralateral, will be located on the left side of the motor. So distinguishing between left versus right could be easier than distinguishing between left versus foot. So in our example, we will be working with left-hand motor imagination versus right-hand motor imagination data. Our first step is to segment the EEG from continuous into trials. Okay, so first we need to define the trials dictionary in order to store the trials of each class. And then we also have to define the time window in samples, okay, to extract for each trial. The time window is, in this case, in the order of seconds, right? So 0.5 to 2.5 seconds. Let's run it. So number of samples will be equal to number of time windows. We use a sampling rate to identify. We have 200 samples, right? So now we can loop over the classes using event codes, event onsets, if the code, and then class. Okay, this is a class label. Okay, left, right, and extract. Okay, we allocate first the memory for the trial in our dictionary and extract each trial from our EEG, right? So EEG, this is our EEG shape, continuous data set, 59 channels, this much, that many samples, time points. Okay, and then we will obtain the trials for two classes, left, right, okay, and then foot. But yeah, this one is right and left hand movement actually. So this is how we defined the segmented EEG. Since the feature we are looking for is a frequency feature, which is related to a decrease in mu activity in EEG, we have to plot or analyze the power spectral density of the EEG trials. In this code, we define a function that computes the power spectral density for each trial. Okay, we will need this later on. We can simply loop through each trials using the power spectral density function, mlab, which uses Welsh method. So for the documentation, we can quickly check. So power spectral density by Welsh average periodogram method. We can read the documentation here. We can now apply the power spectral density on all trials and then store it as the following dictionary. Using the function here, we can plot the power spectral density, okay, that are calculated previously. Because the plotting of 59 channels will clutter displays, we can take the indices of the desired channel, okay, as input and some other metadata to decorate the plot. So let's select three channels, C3, CZ, C4 from these locations, central left, central central, okay, central right, and then run the following function. Then we can see that for three channels, select the three channels, okay, we can see the power spectral change for both classes across all trials. Left, right, left, right, left, right. We see some suppressions and differences in the features. Maybe I can open it like this. Yes. So we can see the changes between left and then right. These differences will be used as input features to our classifier. In other words, we can see mu activity on each channel for both classes. Again, at the right hemisphere, the mu for the left hand should be lower than the mu for the right hand movement due to the event-related desynchronization. As we can see here graphically, the left is less. At the left electrode, the mu for the right hand movement is reduced around these 8 to 12, right? We are interested in these band. And the central electrode, the mu band is about equal for both classes. This is in line with the theory that the right hand movement or right hand movement is controlled by the left hemisphere, and then left hand movement is controlled by right hemisphere, and the leg or a foot or feet movement are controlled centrally. Our eventual goal is to classify these data, right, for BCI control. We will use a machine learning algorithm to construct a classifier model that will distinguish between right hand and left hand movement of this given data. In order to do this, we need to first apply some steps. In other words, first we need to find a way to quantify the amount of mu activity that is present in each trial. Second, make a model that describes the expected values of mu activity for each class. And then finally test this model on some unseen data to see if it can predict the correct class label. We will follow a classic BCI design by blankets described in this paper. The authors use the logarithm of the variance of the signal in a certain frequency band as a feature for the classifier, which has take the logarithm, right, of the on top of the variance of the signal again in mu band. So first we need to define our bandpass filter as follows, which allows us to apply bandpass filter to our signal between the frequency band of interest. So defines low, high pass parameter. We pass the trials along with sampling frequency rate, and the IIR filter is applied infinite impulse response filter. Here we can apply the bandpass filter on class 1 and class 2 data, and we have the following bandpass frequency cutoff frequencies: 8 Hz, then 15 Hz. We can now plot the power spectral density of resulting trials in order to see the suppression frequencies outside the pass band of the filter. Okay, after applying bandpass filter, we will see more clear features. Let me open up this, right? So we defined the cutoff frequency of our bandpass filter as 8 Hz and then 15 Hz. That's why we don't see any other spectral features outside the frequency band of interest. And we can see suppression, increase of the mu band. Next, as a feature for the classifier, we will use a logarithm of the variance of each channel, which here can be calculated using the following function: logarithm of the variance. We are using numpy, variance, to calculate the variance of our input data and trials along the channel axis, and taking the logarithm. The following step extracts the logarithmic features. This, interestingly, will result in a single variable for each trial, right? The dimensionality will be as follows: 59, which corresponds to the number of channels, and 100. So 100 is the number of trials. So each point, each one of the dimensionality of this 100 corresponds to one trial. So variance, log var, is just one number we are using as input feature. Now we can visualize those log var of each channel as a bar chart, those single numbers, using the following plot logarithm of the variance of each trial across all channels. So if you do that, let's see. All right, here we go. We obtain these. The bar chart that shows the logarithm of the variance for left and then right. Can we see some differences? In the values here. Those differences are very small, but there is some difference between two classes as we can observe here. However, we need to maximize the difference between left and right class. So the next step will be to go from all 59 channels to only a few channel mixtures. That will provide us maximum variance. We will use a very celebrated algorithm called common spatial patterns to calculate the mixtures of channels that are designed to maximize the difference in variation between two classes. These mixtures will be called as spatial filters. Basically, we want to maximize the variance between two classes. To calculate the common spatial patterns, we need the covariance matrix of each trial, which we define here as a function, and we also need to perform singular value decomposition for whitening. So we will use NumPy's linear algebra module to perform singular value decomposition on the covariance matrix sigma. All right, and then this is the common spatial pattern will be defined within the following function, CSP in short, which takes trials, let's say from right-hand imagination, left-hand imagination, and then calculates a covariance matrix and whitening and singular value decomposition, and take the inner product, okay, between P and B, and return the weight. So the W will be the spatial filters that will maximize the variance for one class, minimize the variance for another class, for the second class. We can apply this W to our trials using apply_mix method, which basically performs the inner product between each trial and then the weight. Okay, then after applying this filtering, spatial filtering, we can observe its effect in terms of minimizing and maximizing the variance. So I run this all define, and we will get first the CSP. Okay, so weight, W weight, so okay, 59 by 59 square weight, and we can apply to our dataset. So to see the result of the CSP algorithm, we can plot the log variance like we did before and see if it helps. Now, instead of 59 channels and their corresponding variance or log variance, we see 59 mixtures of channels, which we also call as components. Each line is component. They are the result of 59 spatial filters, which we apply to the data, the W. This is 59 spatial filter, right? The first filter maximizes the variation on the first class, while minimizing the variation on the second. And then for example, here, the last filter maximizes the variation for the second class, okay, right class, while minimizing the variation for the first class. Again, maximize the variation for the first class, which is left hand, while minimize the variation for the second. While in the last filter, which is located here, maximize the variation for the second class, minimize variation for the first. So the filtering actually, CSV, common spatial filtering, is based on that. Maximize, minimize the variation in the data. These changes can also be visualized as a part of pulse spectral density. Okay, let's see. Yeah, before moving on, let me open this figure. Okay, here the variation, the difference between left and right is very small. While after applying CSP, we have the following effect. Well, here these are not channels, but features, log variance features or components. So if we visualize the pulse spectral density after common spatial filtering, we will see the following. Okay, open. Well, here. So PSD for right hand imagination class, PSD power spectral density for left hand imagination. And if you compare with the following, you see very significant difference, right? Huge difference. That is the effect of spatial filtering. We are maximizing, we are extracting discriminative features using spatial filtering for two classes. We can now feed these features to a classifier to obtain a good accuracy, classification accuracy. So it is useful to see how well we can differentiate, discriminate between two classes as a scatter plot as well. Okay, here, okay, after running these, we will obtain the following scatter plot between two classes. Okay, on two-dimensional plane. X-axis is the first component, CSV component. Y-axis is the last CSV component. Okay, so first component and then last component. And then we are visualizing it as a scatter plot. Can we see? Can we draw a line? Can we use a linear classifier to find the decision boundary between left and then right hand imagination? Now, finally, once we have these kind of features, we can feed the data to the classifier. We can apply linear classifier to this data set after so much pre-processing, cleaning, making it separable. And again, linear classifier can be thought of putting or drawing a line between two classes of these data, right? Just finding a line that separates them. Okay, and then, yeah, after finding the line, we can check the classifier model on a new trial. Okay, if it will be on the correct side of the, let's say, decision boundary. So in classifier training, as we know, the first step is splitting the data into train and test set. Also validation set, right? The classifier will feed the model, okay, on the training set, and then use this trained model to make future prediction about the test set. We can check on which side of the line each test trial will fall. Okay. Also, here we have a chain of the steps we use for pre-processing the data, which includes a common spatial pattern algorithm as a part of the model, right? Because a CSP is a supervised learning algorithm. Okay, so common spatial patterns can also be thought of as a machine learning algorithm because it uses the labels of two classes to maximize the variance for one class and then minimize the variance for the other class. So it is using the labels to find the filters. So it is a supervised learning algorithm, and we should use CSP with the linear classifier to supervised learning algorithm in chain. Well, for model selection, we can start with the 50 and 50 split ratio here. So training data, we allocate 50%, and then for test 50%. Of course, this is not the correct way of doing some model selection. We usually follow the 80% for training, 50 for testing. In this case, we just want to check, okay, for demonstration purposes. So let's define the number of training for, let's say, right hand and then foot movement, and number of training for, say, right and left is defined as follows. And finally, we use this is test train, right? We use these segments of data to define our dictionary, which will define the training and then the testing. We apply filtering, bandpass filtering, and then we apply the common spatial patterns on train class 1, train class 2, right and then left hand imagination. We can check the dimensionality of our data as follows: 59 channels, 200 time points after applying bandpass filtering, and then 50 examples for training. And then the shape of the weight vector, spatial weight vector obtained from CSP, is equal to 59 to 59. Right now we can apply, okay, the weight of spatial filtering on our train and then test samples, right? For class 1, class 2, class 1, class 2, we will obtain the following pre-processed or spatially filtered data set. Now the shape remains same. You can see that, okay, we can compute the components, or we can just select the first and then last components of our feature, right? Because those are the ones that provide the maximal discrimination, as we can see here, first and then last one. All right, now let's see. So we have only two components, just two components, right? Two features. We can apply logarithm on those components, okay, and then see if how it reduces. Okay, two components, okay, on 200 time points, right? Just one. Then we can see it as follows: two components, 59 channels. So once we have these features, we can finally train our classifier. So for classifier, we can use linear discriminant analysis, LDA. Okay, it's based on fitting a Gaussian distribution to each class of our data, okay, which is characterized by mean and covariance, and also determines the optimal separating plane to divide the two. Yeah, so let's define the LDA also using NumPy's module, okay, which is very easy. And first, okay, train, apply LDA. We have two functions. So first we train LDA on the features that we have extracted. Train class 1, class 2, and we obtain the weight and then the bias. So weight is two-dimensional vector. Bias is a single value, right? So it's much simpler than you can think. It can be informative to recreate a scatter plot that we have seen. Okay, one of these scatter plots, and then plot the decision boundary using the classifier we have obtained. Right? In this case, look, this is plot scatter plot. Let me open these. Okay. Okay, here the decision boundary is this line for which the classifier output exactly 0. All others, okay, can be plus minus. The scatter plot here, okay, used x0 as x-axis and then x1 as y-axis, two features. We see that most of the points are classified correctly, unless except these ones, except few of them. We're using linear classifier. Now we can see the decision boundary on the scatter plot when we use the test data point. Okay, so here we included we use the test points. The difference between two are significant. So on training and test has a lot of more mistakes. More points are misclassified. Once we train the LDA, we can apply it to the test data and present the result as a confusion matrix as follows here. So in PRA, this is confusion matrix. We apply LDA on test class 1, okay, for first and then second class and sum. Apply class 2, class 2. And we can also calculate the accuracy. So the number here at the diagonal will be the trials that were correctly classified. Okay, any trials incorrectly classified will be either false positive or false negative, which will occur at these corners. We can see that from the confusion matrix that 4 out of 50 trials with the left-hand movement were incorrectly classified. Okay, and 5 out of 50 trials with right-hand movement were incorrectly classified here as left-hand. So in total, okay, the accuracy is 91%. This tutorial should be useful to get started with your BCI dataset acquired from Motor-Imagery experiment. CSP, the common spatial pattern or the algorithm, is one of the very popular celebrated algorithms in Motor-Imagery BCI field. We have seen how we can apply this CSP algorithm today, which requires calculating the covariance matrices, log variance, and other necessary steps. We also used the linear discriminant analysis, another commonly used classifier for Motor-Imagery tasks. I hope it was useful. Let me know if you have any questions.